So in this lecture, we're going to move on and talk about Chapter 8, looking at microbial genetics. So we'll start by a few terms. So first of all, what is genetics? Genetics is the study of genes. And there are several components when studying genetics. The first would be what they are, so identifying where the genes are located in the genome, how they carry information. So what does that gene, what does that DNA sequence code for? How is information expressed? Because if we look at an organism's total genome, looking at all of their genetic information, only a small percentage of that genome is actually being expressed. About 1% is what's actually being expressed. And so we need to understand how is that information expressed, meaning when is gene expression turned on, when is gene expression turned off, etc. How genes are replicated and passed on. So understanding how the genetics are passed down from generation to generation. Also, we're going to look at what's called horizontal gene transfer, meaning not just from replication from one generation to another, but also how organisms can share genetic information. So let's start with what a gene is. A gene is a segment of DNA coding for a functional product. Historically, we used to think of a gene as being a segment of DNA that codes for protein. But what we're actually finding is that not all DNA codes for protein. There are some DNAs that code for types of RNA, and those RNAs don't form proteins necessarily, but they do play an important role in the cell. And so, again, when we talk about gene, a segment of DNA that codes for a functional product. So it could be a protein, it could be RNA, it has to be some sort of functional product. Usually, one gene codes for one protein. Now, there are some exceptions to this, but as a general rule, usually one gene codes for one protein. And E. coli has about 4,600 genes. This number is constantly changing, and so estimates vary from time to time, but 4,600 is the amount that's listed in a textbook. Homo sapiens, which are humans, have about 20 to 25,000 genes. And so, interestingly, if you compare humans with Arabidopsis, which is the mustard plants have more genes than humans do. Yet, we are considered to be more complex. And one of the things that we'll learn is that humans, for example, can do something called alternative splicing. They can take one gene and cut it in multiple different ways to give more than one protein from that one gene. So I studied a protein in grad school, and this protein was a transcription factor, which means that it's a protein that regulates which genes are turned on or off. And actually, the gene that I studied formed 3 functional proteins. And interestingly, when we looked during development, those proteins are expressed during different times in development. And they played very different roles in regulating transcription depending on when they were expressed during development. And so while we say usually one gene codes for one protein, know that that's not always true. There are some exceptions to that. Humans, again, do alternative splicing. And later on, when we talk about how transcription and translation work, you'll start to see why E. coli don't do this alternative splicing. There's a reason that prokaryotic cells don't do this. So we need a few more terms. And the next term is a chromosome. And a chromosome is a structure made of DNA that contains the genes, meaning it's used to carry the hereditary information. And so if we look at this diagram on the bottom here, what you're going to see is that you're looking at a linear chromosome in this case. So if we see a linear chromosome, is that chromosome eukaryotic or prokaryotic? And the answer is, if it's linear, we're looking at eukaryotic DNA, right? Prokaryotic cells like bacteria have circular chromosomes. And in the case of E. coli, it has one circular chromosome. If you compare that to humans, let's say, humans have 46 chromosomes. 23 came from mom and 23 came from dad. And so these chromosomes are these long linear DNA molecules, and they contain these genes. Remember that a gene is a segment of DNA that codes for some functional product. And so on this chromosome, we're going to have a variety of genes that code for a variety of different functional products. When we look at a genome is all of the genetic information in the cell. The first organism to have its genome mapped was E. coli. The human genome was completed in 2003. So all the way back 17 years ago in 2003, we had mapped the human genome. Now, just knowing the sequences, though, is not enough, because that doesn't necessarily tell us what those segments of DNA do. And so a lot more work has gone into studying after, now that we know these sequences, what do these sequences do? What is their function within the cell? If we talk about genomics, genomics is the molecular study of genomes. So again, basically the sequencing of the genome, for example, etc. And then we have genotype and phenotype. Genotype is the genes of an organism, meaning which genes they have. So if we think in terms of humans, our genotype, for example, let's say I oversimplify and I look at eye color. Now, eye color is much more complicated than this, but for the purpose of this example, we will simplify it. So let's say we're talking about eye color. Your genotype would be which genes you have. Now, remember that for your chromosomes, 23 came from mom and 23 came from dad. So your chromosomes are arranged in pairs. So for each gene, for each gene, you have two different genes that you can have. You could have two of the same genes, you could have two different genes, but your genotype would have two genes. So if we go back to the eye color example, you could have a genotype where you have one brown-eyed allele. Allele is just an alternate version of a gene. So again, eye color would be the eye color would be the gene, and the allele would be the variation in that DNA sequence that gives us a different outcome. So one DNA sequence could code for brown eyes. One DNA sequence could code for blue eyes. So your genotype would be that you have one brown-eyed gene and one blue-eyed gene. That's your genotype. It's what genes you have. Your phenotype, think of the physical characteristic. And that is, it's the expression of the genes. And what that means is that if my genotype is that I have brown eyes and blue eyes, so I have two alleles, two genes, my phenotype is based on that genotype, based on what genes I have, what is the physical consequence? What do we actually see as a result of having that genotype? And so the phenotype in this case would be that if a person inherited one blue-eyed gene and one brown-eyed gene, their phenotype would be that they have brown eyes. And so again, this is oversimplified. Eye color obviously is controlled by more than one gene because we don't have just brown or blue. We have so many variations in between, which tells us that there are multiple genes that control eye color. But that example was just to kind of simplify and help you understand the difference between genotype and phenotype. So think of genotype, genes. What sequences does that person have? Do they have a brown-eyed gene and a brown-eyed gene? Do they have two blue-eyed genes? Do they have a brown-eyed gene and a blue-eyed gene? And then the phenotype is what we see as a result of having a particular gene or combination of genes. And so that's genotype and phenotype. So what you're looking at here is an E. coli, and this E. coli has been chemically treated. And what happened is that this chemical causes the DNA to come out. And so what you're looking at is all of this yellow strand yarn looking parts are actually the chromosomes, or the one chromosome that's found in that E. coli. And notice that if you look at this chromosome looks so much bigger relative to the cell. The chromosomes, if you were to lay it out flat, the DNA is actually 1,000 times longer than the cell itself. Yet, that chromosome can package up very tightly, and inside of the E. coli, it only occupies about 10% of the space inside the cell. And so what that tells you is that this chromosome is able to pack in very tightly. It's able to coil. It does something called supercoiling. And so it coils and it wraps around and it packages in very, very tightly. If you were to look at human DNA, so human DNA, if you think about your cheek cells that you guys looked at, for example, right? Your cheek cells, you had to use a microscope to look at it. However, if you were to take the DNA out of just one single cheek cell, so one single cheek cell that's microscopic, if you were to take all the DNA out of that one cell and you were to line it up end to end, that DNA would reach about 2 meters or about 6.5 feet in length. So think about that for a minute. Inside your microscopic cell, if I were to put the DNA end to end, it would be taller than I am inside my microscopic cell. If you were to look at all of the DNA in an adult human, and that's about 100 trillion cells or so, if you were to take all the DNA in your body and put it end to end, the DNA would stretch 113 billion miles. To give you an idea of how massive that actually is, that would take you to the sun and back 610 times. So think about that for a minute. Just think of the scale of this. All of the DNA in your body, put end to end, the DNA would stretch to the sun and back 610 times. That is massive. The DNA packs in very, very tight. The human genome is about 3 billion base pairs in one cell, so it's that many base pairs along the sequence. If a person was typing the human genome and could type 60 words per minute, 8 hours a day, it would take about 50 years to type the human genome. So think about that. If you can type 60 words per minute, and you do this 8 hours a day, it would take 50 years to type the human genome. If written out, a human genetic code would fill the pages of about 200 1,000-page New York City telephone books. Now, we don't use telephone books anymore, but your parents might recall using telephone books. If you're older, I actually did use telephone books at one point. But if not, you can still get a scale. So if you think about how large a telephone book, let's say we're talking about New York City, and it's a 1,000-page book, your human genome, if typed out, would be 200 of those 1,000-page telephone books. That is massive. And so that's just to give you an idea of the scale of this. So the DNA packs in very, very tightly. Now, if we look at the E. coli chromosome, remember that the E. coli chromosome is going to be circular. They have one circular DNA. And what you'll notice is this is base pairs. So BP is referring to base pairs. So that's one pair of bases. So like an A paired with T would be one base pair. If we look over here and we see KBP, that's kilo base pairs. So kilo means 1,000. So what you're looking at is that the genes that are found on the E. coli chromosome are color-coded. So these blue ones are the genes for amino acid metabolism. So you can see that we have some genes here, and some here. If we look at the red ones are carbohydrate metabolism. So those are the DNA sequences that are used for carbohydrate metabolism. So here we have one, another, and there's another. If we look at the pink ones are genes for DNA replication and repair. And so notice that to an extent, these are kind of clustered on the chromosome. There's another one out here, but we have this kind of cluster of genes that are used for replication and repair. Lipid metabolism, so we have a cluster of genes here. If we look at membrane synthesis, notice that those are very clustered. They're all found within a short segment on the chromosome of E. coli. Now, in humans, some of our genes are clustered too. And so they're arranged in a cluster and they do something similar, meaning they code for similar types of proteins that have some coordinated function. And so same idea here. So in the E. coli, we have this little cluster of genes that all play a role in membrane synthesis. Now, you might wonder, what are these numbers that are on the inside of this diagram? And what that is that it takes about 90 minutes for the chromosome to transfer from a donor to recipient. And what that means is that if you recall back to when we looked at chapter 3 and we looked at prokaryotic cells. Prokaryotic cells, remember, some cells can have a structure called pili. And pili is a structure produced by some bacteria that allows bacteria to do conjugation, meaning that two bacteria can hook up and they can exchange genetic information. And so what that means is that depending on the amount of time, so let's say that cells had only hooked up for 10 minutes. That means only these genes here would have been transferred. If we get to 20 minutes, it would be all of these. It takes about 90 minutes or so, approximately, to transfer the entire bacterial chromosome. So those cells would have to be hooked up, and that would allow the bacteria to transfer its genetic information from one cell to the next. And so this is, again, you don't have to memorize this map of the E. coli chromosome, but it's just to give you an idea of kind of what does that chromosome look like and where are these genes found. So throughout this lecture on genetics, we're going to break this down into 3 parts. The first part, in terms of the flow of genetic information, is we're going to look at what's called vertical gene transfer. And so that means going from a parental cell, and that cell is going to replicate and give rise to 2 daughter cells. So the first part of the genetics lecture is going to be focused on looking at DNA replication. So how DNA is replicated, how that's passed from one generation to the other. The second part of this lecture is going to be looking at gene expression. And what that means is now we're talking about the central dogma of biology, meaning how we go from a DNA sequence and we transcribe it to make an mRNA. That's transcription because we're transcribing a similar language. DNA and RNA are both nucleic acids. And then the mRNA is going to code for the protein. And that process is called translation because we're translating. We are going from nucleic acid language to protein language. Those are different types of macromolecules. And so this is translation. And so we're going to look at how gene expression occurs within the cell. How does the cell make those proteins, which give us the phenotype? Remember, the phenotype is the physical consequence of having a genotype. So depending on what genes you have, that will dictate what proteins get produced. And if you think about all of the biochemical tests that we've looked at so far, let's say we're talking about starch hydrolysis. If we're talking about starch hydrolysis, remember we're testing to see, are bacteria able to hydrolyze starch? Can they break down starch as a food source? And essentially what we're testing is, do they produce amylase? Amylase, ACE enzyme. Enzymes are protein. And so what allows bacteria to either make or not make amylase is depending on, do they have the gene? Do they have the DNA sequence? Because if they have the DNA sequence, then it can be translated to make that enzyme, which allows them to do that metabolic process. And so the genetics of it all is ultimately what dictates which metabolic pathways different bacteria can do. Why some can do sugar metabolism and others can't. It all comes down to enzymes. Which enzymes do bacteria have? Which ones do they not have? And again, that's all going to come down to genetics. Which genes the organism has. And so the second part of this lecture is going to focus on gene expression. So how do we go from transcription and translation? How does the cell process that information? And then the last part is going to focus on what's called recombination. And that's going to be our horizontal gene transfer. And so that's going to be a transfer of genetic information between two cells of the same generation. So for example, we'll look at a type of recombination that we've already talked about, which is conjugation, right? One cell can form sex pili, connect with an adjacent cell, and pass DNA from one cell to the other. And so that's going to be our recombination. And so the third part of this lecture will be looking at recombination and talking about how recombination occurs, what types of genes can be transferred along this line, etc. So, biological problem. Where does the cell's DNA go when the cell divides? So if you think about it, if you have, let's say, a cell and it has some genetic information. If that genetic information does not duplicate before the cell divides, you're going to end up with one daughter cell that has DNA and the other one does not. And so the biological solution to this is that the cell is going to replicate its DNA first. So it's going to take its DNA and it's going to make an exact copy. And then those two copies are going to split into two cells. And these daughter cells are now offspring and identical copies of this parent cell. And so bacteria, for example, will do this through a type of asexual reproduction. Remember that they are a single-celled organism. They exist as one single cell. So when they reproduce asexually without a partner. They simply just copy their DNA. Remember that they go through a process called binary fission, and they will divide into two daughter cells, both of which are identical copies to the parental cell. So the structure of DNA was published in a model in 1953 by two scientists by the name of Watson and Crick. And so Watson and Crick published a paper basically proposing that DNA is a double helix. Now, their model used several facts to decipher that it's a double helix. And the first is that DNA is a linear polymer of 4 types of bases linked by sugar phosphate groups. So at the time, scientists had already discovered that there were 4 types of bases. There was A, G's, C's, and T's. And they knew that it was sugar and phosphate groups. So they knew that it was sugar phosphates, and they knew there were 4 bases, but they still weren't sure about what the overall structure would look like. The ratio of bases they found, not them, but other scientists found, was that A equals T, G equals C. And this became known as Charge-Off's ratio. And that is when they looked at different species of organisms, didn't matter which species they looked at. What they always saw was when they quantitated the amount of A's and T's and G's and C's, that a percentage of A's always equal to percentage of T's. So let's say, for example, that overall when they looked at the nucleotides, they found 30% of the nucleotides were A. That means in that organism, they always found 30% T. Didn't matter which organism they looked at. This was always the same. A equals T. There was 30% A's and there were also 30% T's. Combined, notice that makes up 60%, which means that the remaining 40 was left to G's and C's. And so half was G and half was C. So 20% were G, 20% were C's. So G equals C. And so again, no matter what species, no matter which organisms they looked at, A equals T, G equals C. And so they said, well, why is it that A is the same percentage as T and G is the same percentage of C? What does that tell us? And so the last piece of the puzzle that they needed in order to solve their model of DNA was that they figured out that DNA is a helical structure with a helix width and base pair spacing that was known from work by Rosalind Franklin. And so Rosalind Franklin was a female scientist, and she did a technique called X-ray crystallography. Now, this is not an easy technique. She had to crystallize DNA and then subject those crystals to X-ray. And when the X-ray would hit the DNA crystals, the X-rays would diffract and they would produce an image on film. And this is what she got. So when she did this and she took that crystal and she subjected it to X-rays and then put film behind it, she got this image. Now, I did have the opportunity to see Watson talk when he came to UCI, and I will just say in this recording that he had some very interesting things to say on the matter. If you want to ask me more about that, you can. But basically, what he basically said in regards to Rosalind Franklin's data was that they basically, without her permission, they saw this image in her lab, and they basically stole her data. They saw this piece of film, and this was kind of the final piece of the puzzle that basically told them that this structure is a double helix. Now, I look at this image and I would not have known what this meant, but this X is this double helix. And so when they saw this image, that was kind of their final piece of the puzzle. That was the last piece of information they needed to publish that that DNA is a double helix. So Watson and Crick were given the Nobel Prize for this discovery. Unfortunately for Rosalind Franklin, even though her work played a big role in the model of DNA, Rosalind Franklin died before they received the Nobel Prize. And they don't give Nobel Prizes for people who have already passed on. And so poor Rosalind Franklin never got really the notoriety that came with discovering that structure. You have to think that during this time, she was working with X-rays, and they didn't quite know the extent of how dangerous X-rays were, what were good precautions to take. And so as a result of working with these X-rays, she ended up getting cancer. and she died. And so Rosalind Franklin really never got kind of the fame that she should have gotten, or the acknowledgement that she should have gotten for this discovery. Watson and Crick basically got all of the glory. And you're more than welcome to look up Watson and the crazy things he said over the years. It's pretty outlandish. And again, if you want to ask me more about it later, you can. So I have a little review for you, just to kind of review about our macromolecules. So what I'm going to do is I'm going to read these out. And then what I want you to do is I want you to pause, try and answer these yourself, and then I will go over the answers with you. So let me read them first. DNA and RNA are blank. So what type of biological molecule are they? What's their class of macromolecules? The monomers that make up DNA and RNA are called blank. So what do we call the building blocks? The polymer is called blank. And DNA is an abbreviation for blank. So again, pause your video. When you're ready, push play, and I will go over the answers with you. Okay, so hopefully you attempted this. DNA and RNA are what type of biological molecule? They are nucleic acids. And the monomers that make up DNA and RNA are called nucleotides. Right? The building blocks are the nucleotides. And recall that back when we talked about our nucleic acids, when we looked at nucleotides, remember that nucleotides have 3 parts: a sugar, a phosphate, and a nitrogenous base, which we'll talk about more in a minute. The polymer is called, remember from our table, nucleic acids. So nucleic acids, you could also refer to them as polynucleotides. That is technically right as well. Either could be used here. And then DNA is an abbreviation for deoxyribonucleic acid. So deoxyribonucleic acid, DNA. And then RNA is ribonucleic acid. So it doesn't have the deoxy part. And so again, we'll talk about differences between DNA and RNA in just a minute. So again, just to review, building blocks of DNA, again, are referred to as a nucleotide. And each nucleotide has 3 parts. It has a sugar, and the sugar is deoxyribose in DNA, or ribose in RNA. So we have our pentose, our 5-carbon sugar. So carbon 1, 2, 3, 4, and 5. And again, the deoxyribo comes from the fact that at carbon 2, there's no oxygen on that carbon. It's just a hydrogen. If you were to look at ribose, that would have an OH group at carbon 2. It has the oxygen. So we have our pentose sugar, our 5-carbon sugar. We have our phosphate group. We have 1 phosphate group. So here's our phosphate group. And our phosphate group is on what we call the 5' carbon. It's the carbon that's in the 5th position relative to this oxygen. So that's why I've numbered this carbon 1, 2, 3, 4, and 5. So we have our 5' phosphate group. And our DNA is going to contain, our nucleotide for DNA is going to contain 1 of 4 bases. A, adenine, G, guanine, C, cytosine, or T, thymine. And so it's going to have 1 of those nitrogenous bases. Again, if we're talking about RNA, it's going to be A, G, U, because RNA doesn't have T, or C. So that's for RNA. And so this is the building blocks of the DNA. And so the next video is going to focus on the overall structure of DNA, and it's going to pick up where this video leaves off.